All right, so hi everyone. So like I mentioned in the main session, so during these lightning talk sessions, each presenter will speak for 3 minutes. And we ask that all audience members hold their questions until the end of all the lightning talks in the session. And at that point, we'll open up the floor for discussion of all the talks. So at this point, audience members can raise their hand and the moderators will call on them and allow them to unmute themselves, and they can ask the speakers questions directly. Audience members can also type their questions into the chat, and the moderators will then pull them into the discussion. So when you're not speaking, we ask that all participants have their microphones muted. And if Audrey, if you could help me monitor the chat, that would be great.
Yes.
So our first speaker will be Mariko Kanai. So a little bit about Mariko. She's a fifth-year PhD student in Dr. David Fidduck's lab at Columbia University Irving Medical Center. And her thesis work harnesses Plasmodium falciparum genetic crosses to investigate the mechanisms of action and resistance to the antimalarials chloroquine and quinine. So take it away, Mariko.
Just one more comment before you start. You'll see that I'm going to raise my hand at 2 minutes and 30 seconds, indicating that you have 30 seconds left. And then once you go over time, I will comment and let you know that as well. Okay, go ahead.
Thank you. Thank you for the introduction. So hi, as Victoria mentioned, I'm Mariko Kanai. I'm a fifth-year PhD student in David Fidduck's lab at Columbia. And today I'll be showing a new Plasmodium falciparum genetic cross that our lab conducted in collaboration with the Deepti Srivastava lab at Johns Hopkins. And so I will be talking about quinine, which is an antimalarial that has been used to treat malaria since the 1630s and is an alternative treatment to artesunate for severe. Several partial mediators have been identified, but none were validated in clinical isolates or by reverse genetics. And in addition, the primary driver of quinine resistance has not yet been identified. So one forward genetics approach to identify mediators of quinine resistance is to utilize the Plasmodium falciparum genetic cross. And to walk you through the process, looking at the diagram, we started with 2 genetically and phenotypically distinct parasites as their cross parents. NF54 is a quinine sensitive African strain, and CAM 3.2 is a quinine resistant recent Cambodian isolate. And then gameticides from NF54 and CAM 3.2 were mixed and fed to mosquitoes. And then sexual reproduction and meiotic recombination occurred in the mosquito midgut, creating new recombinants. And then after the parasites underwent the human liver and blood stages in the human liver chimeric mice, we obtained the falciparum infected RBCs and cloned the progeny with or without drug pressure. So in total, we obtained 478 progeny, which segregated into 112 unique recombinant progeny. These parasites were then whole genome sequenced, and a subset were also assayed in drug assays. So I profiled the progeny using in vitro quinine dose response assays and obtained inhibitory concentrations for 50% and 90% growth over 72 hours, so IC50 and IC90 values. And if you look at the bottom left, quinine response did not segregate with known mutations in PFMDR1 and PFCRT. CRT is a gene known to drive resistance to chloroquine, and both CRT and MDR1 have been previously partially associated with quinine resistance. So then we used a statistical tool called quantitative trait locus mapping, or QTL mapping, which is the bottom middle figure that links genotype such as WGS and phenotype such as drug assay data to look for genomic regions that associate with resistance. And we found that quinine IC90 high-grade resistance associated with a new chromosome 7 peak that did not contain CRT. This chromosome 7 peak only contains 3 genes that have non-synonymous mutations between the parents. And of these 3, the drug metabolite transporter 1, or DMT1, is the most promising candidate, as it is similar to CRT in size and number of transmembrane domains, and is in the same drug metabolite transporter family. And the 2 mutations in DMT1 that we found between the 2 parents have been also found in the field. So we are currently conducting gene editing studies of DMT1 to investigate its role in resistance to quinine and other antimalarials.
Awesome, thank you. That was lovely.
Yes, thank you, Mariko. So we will now move on to our second lightning talk, which will be given by Heather Colvin. So Heather is currently a microbiology and immunology PhD candidate at Wake Forest University in Regina Cordy's lab. Her research focuses on metabolic perturbations in malaria and malarial anemia. So take it away.
Thank you. Hello, everyone. Thank you for the opportunity to briefly talk to you today about an ex vivo model of oxidatively stressed red blood cells and exogenous amino acids in enhancing bystander red blood cell function and morphology in malaria. So malarial anemia is multifactorial, and in the field, there are three main processes that are known to occur to contribute to anemia malaria. As we all know, there's an obligatory destruction of infected red blood cells as the parasite undergoes its intraerythrocytic life cycle. To a greater extent, around 8 to 9 uninfected red blood cells are also lost for every one parasitized red blood cell during malaria. And this phenomenon is known as the bystander effect and is not well understood. Also, in addition to the loss of infected and uninfected mature red blood cells, the production of new red blood cells is affected and dyserythropoiesis occurs during malaria. The work I want to highlight to you today focuses on the bystander effect and potential route for lessen this phenomenon metabolically. Our previous work found a decrease in plasma metabolites in acute malaria, which is also when there's a lower hemoglobin or anemic condition during malaria. So one of these significantly decreased plasma metabolites found in vivo was L-glutamine. Decreased plasma glutamine was really interesting to us because oral glutamine powder is an FDA-approved treatment for sickle cell disease. And while sickle cell is not a result of a parasite, but as malaria is, but malaria and sickle cell both are highly oxidative in diseases that involve anemia. The diagram on the bottom left of the screen highlights active metabolic pathways within a red blood cell. And as we can see, exogenous glutamine can be taken in, converted to glutamate within the cell and used as a precursor for glutathione synthesis, which is a crucial antioxidant within the cell. In addition, exogenous cysteine and glycine are also needed in this pathway. So to begin looking at this, we created an ex vivo oxidative stress red blood cell model by stressing red blood cells from a healthy donor with various concentrations of hydrogen peroxide, a known oxidative stressor. Shown here are scanning electron microscopy images of red blood cells after hydrogen peroxide stress, and we can see the morphological presence of echinocytes increase as hydrogen peroxide concentration increases, which we expected to find based on literature. So to begin to more understand the role of exogenous amino acids in this model, we added them either singularly or in combination to our stressed red blood cells and found that, as shown in the left graph, the addition of all three amino acids, glutamine, cysteine, and glycine, as well as glutamine and cysteine alone, improved the presence of normocytes, or this normal bioconcave morphology we all recognize as a red blood cell. And then furthermore, using ectocytometry, which is a method relying on osmolality gradient and laser diffraction to measure red blood cell membrane properties, we saw that in the third column that hydrogen peroxide stress alone decreased red blood cell deformability, meaning that the red blood cell membrane was more rigid. However, the deformability was significantly improved in the population of red blood cells with exogenous glutamine, cysteine, and glycine amino acids. And this would mean that overall, those red blood cells were less rigid and more flexible in the presence of these amino acids. We also saw that a significant increase with cysteine alone. Altogether, our oxidatively stressed red blood cell model supports our hypothesis that these exogenous amino acids can help the red blood cell membrane, and we are currently working on looking at this in the context of bystander red blood cells from Plasmodium falciparum culture. Thank you, guys.
Great, thank you so much. That was very interesting. Okay, so our third lightning talk presenter will be James Mcclellan. And James Mclean is a fourth-year PhD student in Kirsten Hansen's lab at the University of Texas at San Antonio. He's currently working to develop novel assays to power drug discovery and investigate the cell biology that drives syncytial growth and development in Plasmodium liver stages. So, floor is yours, James.
Thank you so much. So Plasmodium-specific translation inhibitors are a desired class of antimalarial compounds. But currently used translation assays rely on population-level readouts in P. falciparum asexual blood stages. So in order to assess single parasite responses to translation inhibition, we developed a quantitative bioimaging assay in the liver stages using P. berghei infected HepG2 model. So this assay uses O-propargyl pyrimycin, or OP, to label the nascent proteome of infected cells, and click chemistry provides a visual snapshot of the total translational output, here labeled OP A555, as demonstrated in the DMSO images in the top left. So we assess the specificity of this labeling in acute pretreatment with cyclophosphamide. This blocks the signal in both host and parasite, while the Plasmodium-specific inhibitor DDD498 only blocks labeling in the parasite. The specificity allows us to quantify the effects of translation inhibition independently in parasite and host, as shown in the box plots below. So our quantification strategy uses automated confocal feedback microscopy to generate image sets of single exoerythrocytic forms, or EEFs, and batch segmentation and feature extraction separates OP-A55 fluorescence associated to EEFs, shown in green, and corresponding in-image HepG2 in orange. This metadata-rich approach links single measurements to their associated image. So we assembled a set of pan-eukaryotic bioactives, including known translation inhibitors, compounds that do not target translation mechanistically, and various antimalarial compounds to assess the sensitivity and specificity of this assay using an acute pretreatment prior to a 30-minute op labeling. So 10 of our 19 test compounds caused a 50% decrease in translation. We confirmed this in concentration response. So all 8 of our known translation inhibitors validated, including the Plasmodium-specific inhibitor DDD498. The box plots on the left demonstrate differences in potency and selectivity. So nisomycin is nearly equipotent between parasite and HepG2. Presantin is parasite selective at submaximal concentrations. And DDD498 is completely parasite specific. So nisomycin and DDD498 also cause homogeneous inhibition of single parasites at active concentrations, while parasite response to presantin treatment is highly variable between individuals at a 1 nanomolar concentration. So we identified 2 surprising compounds that cause translation inhibition in the acute assay, the well-known proteasome inhibitor bortezomib and the antimalarial CAF156, which is believed to work at the level of the parasite ER. So even though these compounds validated in concentration response, it does not necessarily mean they're targeting translation directly. And an advantage of this assay is flexibility in pretreatment times. So we forced the compound and OP label to compete for inhibition of protein synthesis directly. In the plot at the bottom right, we see that bortezomib and CAF156 lose activity altogether, indicating an indirect shutdown of translation in the acute assay, while critically, all of our known translation inhibitors caused identical levels of inhibition in both assay modes. So in summary, we've developed an image-based assay to separate and independently quantify translation inhibition in P. berghei liver stages. Flexible treatment times differentiate between direct and indirect inhibitors, and we hope this will be useful in the context of drug discovery. Thank you.
Thank you so much, James. That was really interesting. All right, so moving on to our next speaker. So our next speaker will be Frank Addae. So Frank Addae is a scientist from Ghana who recently completed his undergrad studies in biochemistry, cell, and molecular biology at the University of Ghana. So his research interests include infectious diseases and drug discovery. The floor is yours, Frank.
Thank you very much for the kind introduction. So the malaria parasite has 2 hosts, the female Anopheles mosquito and the human host. In the human host, the parasite is highly synchronous. However, in vitro culturing, they are asynchronous. Now, to carry out various downstream studies on the parasite, they need to be synchronized to mimic what happens in the human host. And various synchronization methods, such as the use of 5% sorbitol solution, the use of PECOL and magnetic separation, and other methods have been established to serve that purpose. Even though these methods have been used over the years, not much is known about how they affect the responses of malaria parasites to antimalarial compounds. This study therefore evaluated the effects of sorbitol synchronization of clinical and lab isolate of P. falciparum on their susceptibility to antimalarial compounds. To do this, both the clinical and lab isolate were cultured until more than 5% of the ring stages were obtained. The parasites were then synchronized using 5% sorbitol solution, and the parasitemia was determined using GEMSA stain and microscopy. The parasites were then subcultured for 48 hours. Then growth inhibitory assay was performed using 5 antimalarial compounds, which included artesunate, chloroquine, and 3 drug-like compounds from the malaria box. And this was done on a 96 well plate, with each of the well having a total volume of 100 microliters, consisting of 10 microliters of the compound and 90 microliters of the parasite. The assay, after being gagged, was incubated for 72 hours. And after 72 hours of incubation, the assay was stopped using 80 microliters of licensed buffer containing cybergreen. It was then incubated for 30 minutes in a dark room at 37 degrees Celsius. After that, the parasitemia was quantified using Varioscan last multimode plate reader, and GraphPad present version 9.0 was used to estimate the IC50 values of the compound. At the end of the study, I found that sorbitol synchronization actually affects how the parasite responds to antimalarial compounds, saying the IC50 values for both asynchronous and synchronous parasites differed, as we can see from table 1 on my slide. However, even though the sorbitol affects how the parasite responds to the antimalarial compounds, the clinical isolate had varied responses, while the lab isolate showed uniform responses to the antimalarial compounds. So in conclusion, I've been able to identify or confirm that sorbitol synchronization affects how malaria parasite respond to antimalarial compounds. And so when synchronizing both clinical and lab isolate for downstream studies, the variations in parasite behavior arising from the synchronization method or step should be considered. Thank you very much, and I'll be glad to answer your questions and consider your suggestions. Thank you.
Great, thank you so much, Frank. It's really interesting to think about how sorbitol affects our assays. All right, so our next speaker will be Kyle Mclean. So Kyle Mclean is a postdoc in the lab of Joaquin Niles at MIT, and he did his PhD actually with Marcelo Jacobs Loreno here in the Johns Hopkins Malaria Research Institute. So Kyle, the floor is yours.
Hi, I'd like to start by thanking the organizers for giving me the opportunity to share my work. My name is Kyle Jared McLean, and I'm a postdoc with Joaquin Niles at MIT. And in the Niles lab, our go-to approach for studying the biology of Plasmodium falciparum is through transfection. And traditionally, what this means is we build circular plasmids in E. coli and then electroporate them into the parasite. But as I'm sure many of you are well aware, just the process of building these plasmids is often more difficult than it sounds. The extreme AT richness of the falciparum genome, particularly in the non-coding regions, often leads to plasmid instability and cloning failure. The best solution to this problem is to use linear plasmids. And by that I mean plasmids that replicate as a linear species in the E. coli cell itself. This is the technology that underlies the Plasmodium resource for Plasmodium berghei. And it's something we've been using in the Niles lab with great success for over 5 years now. However, the only commercially available linear plasmid is really big and really difficult to manipulate. So what I've done is rebuild this plasmid from scratch. It's much smaller and much easier to work with, and it works great. But we've taken this one step further and implemented a technology that's popular with our colleagues in the synthetic biology community known as modular cloning, or MoClo. And what this entails is building a library of standard reusable parts, things like promoters and selectable markers and epitope tags, and then stitching them together with high efficiency and high reliability in a single reaction. Using this technology, we've been able to build extremely AT-rich, extremely repetitive plasmids of upwards of 30,000 base pairs in size with very little planning and a near perfect success rate. It's fast, it's reliable, and it's completely changed the way we approach transfection projects. So this tool is a resource we'd like to make available to the whole community. So if you're interested in knowing more about the nitty-gritty details of how all this works, or if you'd like to talk about how you could implement this in your own workflows, please reach out to me to discuss things further. Thank you.
Wow, that's a pretty great tool that we'll now have. All right, so our next speaker is Deepti Srivastava. So Deepti is a PhD researcher working at the Central Drug Research Institute in Lucknow, India. She's currently investigating the functional role of MDR proteins in P. falciparum. So go ahead, Deepti.
So am I audible? First
Yes.
of all, Okay, so good afternoon to all. First of all, thank you so much for inviting me. Today, I'll be discussing my ongoing research, and it's not yet over, but let me tell you, I got a valuable results. So let's start. So I am working on one of the MGR protein of Plasmodium falciparum. It belongs to superfamily ABCB, and it's essential in blood stage. And important point is that recently we came across a paper that shows there is a significant association between the number of asparagine residues and it is linked to drug susceptibility. And we have seen people have shown that if the number of asparagine residues are 7 or 9, it is less susceptible to quinine, and if the number of asparagine residues is 6, it is susceptible to lumifantry. And then if we focus upon upon its homologs, that yeast or bacterial, it is also involved in the export of iron or sulfide intermediates from mitochondria to cytosol, and also it is involved in the transport of glutathione polysulfide. So keeping all these points in our mind, so we thought to mine more research biochemically regarding MDR6 proteins. So these are the results we got. First of all, we raised polyclonal antisera, and by confocal microscopy, it was shown that PFMDR6 is targeted to parasite mitochondria, and it is also confirmed by organellar fractionation assay. MDR6 was particularly expressed in the organellar fraction. Then if we move on to the oligomeric profiling of the MDR6 protein, then it is already known that the ABCB superfamily of Plasmodium falciparum are all half transporters except MDR1 protein. So to check the oligomeric status of the protein, we checked it on 6% native page, and we observed that MDR6 is a homodimer that is confirmed by TEM analysis. And further, when we cross-link the PF lysate with with DSP, it's a permeable cross-linker, it was shown that the amount of dimer increases as compared to monomer, that again confirmed the presence of in vivo dimeric form of MDR6 protein. Then in the bottom left corner, here it was shown all as MDR6 consists of conserved glutathione binding sites. So we were into whether is there any role of glutathione binding in the ATP hydrolysis of MDR6 protein. So here, interestingly, we saw that the MDR6 shows twofold glutathione. Then, but if we compare about its homolog, then the ATP hydrolysis is almost twice in the presence of reduced glutathione. So this is an interesting difference that we saw in Plasmodium falciparum. Then further, we saw that the protein was purified in reddish brown form, and also its human homolog, MDR6 human homolog, is an iron sulfur cluster transporter. So keeping that point in mind, we thought whether is there any role of FES cluster in the ATP hydrolysis of MDR6 protein. So here again we saw that in the presence of glutathione and the holo form of the protein is required for the stimulation of ATP hydrolysis of PFMDR6 protein. So suggesting that maybe there is any conformational role played by 4FES cluster in the conformation of MDR6 protein, thereby the ATP hydrolysis stimulated by glutathione, oxidized glutathione. Then further, we were into what could be the interacting partners of the MDR6 protein. So we dig out two of the components of ISC pathway that is already been previously, this work has already been previously done in our lab. So we saw that PFMDR6 CTD domain is particularly interacting with two components of ISC pathway, that is PFISCU and ISCA2. And then the next question that arises is whether MDR6 is able to accept any FES cluster protein from the scaffold protein, from the transfer protein of ISC pathway or not. So here we carried out an FES cluster transfer assay in this graph, and here we saw that MDR6 was able to accept 4FES cluster from the transfer protein of ISC pathway, that is ISCU. So till now,
all right, Deepti, we're gonna have to ask you to wrap it up, maybe like one more sentence.
Okay, so it's like finally this is in the bottom right corner, it's a diagrammatic representation that says that MDR6 might be able to transfer iron and intermediate, sulfide intermediate, glutathione intermediate, or glutathione iron sulfur conjugated intermediate. So till now this has been all done. Thank you for your attention.
Thank you so much, Deepti. Okay. All right, so our last presentation today will be from Laura Hagenauer. So Laura is a PhD student at Columbia University, also in the lab of Dr. David Fedak. Her research focuses on the mechanism of piperacin resistance in Plasmodium falciparum and utilizing that to predict the emergence of piperacin resistance in Africa. So take it away, Laura.
Thank you, Victoria. So as you mentioned, today I will be presenting my research on predicting the emergence of piperacin-resistant falciparum malaria in Africa. In Asia, the emergence and spread of mutant K13 led to widespread artemisinin failure, which put increased pressure on partner drugs in the ACT combination therapies. And so this was quickly followed by mutations in PFCRT that have been shown to mediate piperacin resistance. Piperacin works by inhibiting the detoxification of heme in the parasite's digestive vacuole, and mutant PFCRT can efflux piperacin away from its site of action into the cytosol. So piperacin is currently being evaluated as an ACT partner drug in Africa, particularly for seasonal malaria chemoprevention. So we wanted to ask the question, could piperacin resistance arise in Africa like it did in Asia, especially with the rise of ART-resistant parasites emerging in Africa? Piperacin-resistant mutations usually occur on a mutant PFCRT haplotype, and the main ones in Africa are GB4 and CAM783, which are shown in this haplotype table on the bottom left. These two haplotypes differ from DD2 only at 1 or 2 positions. So we thought that the mutations seen in Asia may confer resistance on an African haplotype. We edited DD2 parasites to express these haplotypes plus one of the 3 mutations shown in this box here. We then assayed the response of these lines to piperacin using the piperacin survival assay. I'm showing the GB4 lines on the left and the CAM783 lines on the right in the top right quadrant. DD2 F145I in purple served as our highly piperacin resistant control. And the only line that exhibited high-grade piperacin resistance was DD2 GB4 F145I. But the GB4 T93S line and the CAM783 T93S line did show higher levels of survival under low concentrations of piperacin. We also assayed our lines against a panel of common antimalarials. In Asia, piperacin-resistant mutations were associated with increased susceptibility to chloroquine, and we observed the same phenotype here with all of our novel edited lines exhibiting increased susceptibility to chloroquine and the primary chloroquine metabolite monodecethylchloroquine. The IC50 values are shown in the bottom right quadrant. We also looked at the response of these lines to another foraminoquinoline drug, monodecethylamidioquine, and we found that the addition of F145I sensitized the parasite to MD80Q, but the other 2 mutations we studied did not alter susceptibility to this drug. So in conclusion, there is the potential for PFCRT-mediated piperacin resistance to emerge in Africa. So in regions where DHA piperacin is heavily used, the PFCRT haplotype should be monitored closely for any of these mutations to arise. Thank you.
Great. Thank you so much, Laura. That was a really great lightning talk panel. I know that we're getting close to 1:30, but if people want to, I will stop sharing my screen. If people want to ask any questions, feel free to raise your hand, and then I'll call on you to unmute yourself.
And there are also some questions that have been asked in the chat that we can read out if no one wants to come up with one. So it looks like Jenny Goulaire from UVA asked about a peak that was on the chromosome 12 in reference to Mariko's first talk. And it looks like Mariko was able to answer that in the chat. But we'll go ahead and answer it for everyone's benefit. So the chromosome 12 peak for the quinine IC90 accounted for about 29% of the phenotypic variances and included 146 kb segment that was also observed in the QTL. Since chromosome 12 peak is large, we were hesitant to mention any specific genes, but we were definitely interested in seeing whether this segment is involved in quinine resistance.
Yeah, another question from the chat. Kritika asks, hi Deepti, very cool finding. Is MDR6 predicted to have an FES binding domain? Deepti, do you want to unmute yourself to answer that?
Yeah. So MDR6 has a conserved FES binding domain, and it is present in the C-terminal part of the MDR6 whole protein. It consists of a lot number of cysteines and histidine, which form 4Fe4S cluster compacted in the C-terminal domain of MDR6. Maybe this gives your answer for your question.
Great, thank you. And then I see another question in the chat, also from Jennifer Guler. Interesting, Mariko, is GCH1 in there? Just curious. Oh, it looks like Mariko answered that in the chat. Mariko, do you want to answer that out loud for everyone?
So this was about the chromosome 12 peak that I didn't have time to mention, that also we found in our quinine IC50 and IC90 QTL. And so this chromosome 12 peak was quite large, 248 kb, and it was around 1.5 million base to 1.8 million base for the quinine IC90. So it didn't include GCH1, but it was definitely interesting that we found some overlaps with the chromosome 12 peak we saw for the QTL for other drugs that we looked at, such as MDCQ, which is the active metabolite for chloroquine. So we're definitely looking into this further to see if it's involved in resistance.
Great, thank you so much. Before we close, we can open it up to any last questions. Does anybody have anything they'd like to ask any of the presenters?
I have a question that I'd like to ask Frank.
Yeah.
About the effect of sorbitol synchronization on the drug sensitivity of these parasites. So I saw a lot of changes in that table of yours about changes in the drug sensitivity, but I was wondering if there's a consistent pattern. I didn't quite have time to look at all of them. Is it consistently, do they become more sensitive or more resistant?
All right, thank you very much for the question. So considering the clinical isolates, like you can see from the table, the responses varied. So you can see that at a point in time, the asynchronous was susceptible to some of the drugs, while at some point to the synchronous was rather susceptible to some of the drugs. That is for the clinical isolate. But then for the lab isolate, the synchronized was actually susceptible to all the drugs. So you can see that after synchronizing the lab isolate, the IC50 values decreased.
Yeah, that's very interesting and makes us wonder what that means for all of our parasites that we're always, syncing and drug treating.
Yes,
very
definitely.
cool work.
I do have a question for Heather Colvin. So you mentioned that you use hydrogen peroxide stress, but in your assays. But can you comment on kind of how applicable that is? Like, does it kind of look like the stress that happens in the human body?
Yeah, that's a good question. So we're using a really high amount of hydrogen peroxide stress, and really we got to that level based on our ECTA cytometry curves, just to have a noticeable difference between our normal healthy red blood cells. So I would not say that it's a physiological level, but more so just to induce a large amount of stress to see what changes are happening as a model to then go and elucidate what's happening in the bystander red blood cells.
Great. No, that.
Have a follow-up to that question, Heather. So you mentioned that you get changes in deformability with that oxidative stress, but do you get changes in deformability at hydrogen peroxide concentrations that would be equivalent to the amount of oxidative stress in the bloodstream? Like, is this causing effects actually in an infection?
Yeah, it's really the amount of hydrogen peroxide within a red blood cell in vivo is hard to quantify due to the constant change in ROS, but at lower concentrations, like a 5-fold lower, we do get significant changes in deformability as well. But in micromolar, it's really, we're using healthy red blood cells from healthy donors, which have intact catalase and glutathione peroxidase, so their antioxidant systems are not stressed out as they would be in diseased patients with like already oxidative conditions.
Wonderful, thank you.
And then does anybody else have any questions? I have one last question for James. You mentioned that you work with translation inhibitors, but can you talk a little bit more specifically about like if they're kind of general translation inhibitors or if they're like specific to certain things?
Right, thanks for the question. We tested a variety of pan-eukaryotic inhibitors. So anisomycin and brusantin, for instance, will affect both HepG2 and parasite. But we also tested a number of Plasmodium-specific inhibitors. So the only one that I showed you today was DDD498. But there are some other inhibitors that act on lysyl tRNA synthetase inhibitors, things like that. And those cause inhibition in the parasite only, no effect in HepG2. Yeah.
Great. No, thank you for that clarification. Does anybody have any other questions? No? Okay. Well, with that, I just want to thank everybody again. I know we ran 5 minutes over, but thank you so much for attending the conference today. We heard some really great research. If you have any questions after the conference, feel free to reach out to an organizer and we can point you to the person that you want to reach out to. So everybody, thank you so much. Have a good rest of your day and a good weekend. Bye.